sequencing coverage and depth. Sequencing depth refers to the number of times a specific base, or you can say nucleotide, in the DNA is read during the sequencing process. In other words, it's the average number of times a given position in the genome is sequenced. And sequencing coverage, which is closely related to sequencing depth but provides a broader perspective. So coverage is the proportion, or you can say the percentage of a genome that has been sequenced at a certain depth. It gives an idea of how much of the entire genome has been effectively read and is usually expressed as a multiple of the genome's size. So let's take an example of human genome, which is around 3 billion base pairs. So it would be sequenced to an average depth of 30x. And that means the coverage would be 30x. Now this implies that on average, each base in the genome has been sequenced 30 times. So in practical terms, the higher the sequencing coverage, the more times the genome is read, resulting in a more accurate and reliable information. Now, why is coverage important? The points are variant detection. So higher coverage generally leads to more accurate variant calling. Completeness. Adequate coverage ensures that a larger portion of the target region is sequenced, thus increasing the likelihood of identifying all relevant genetic variations. Statistical power. So coverage directly impacts the statistical power of downstream analysis, such as identifying genes associated with disease. This was all. Thanks for watching.